how much do we really understand about DNA? Beyond just that iconic double helix image, most of us get that it's the blueprint for life, but what about the invisible forces controlling it? Or, and this is pretty mind-bending, what if genetic information can actually flow backward? Welcome to the Deep Dive. This is where we take your stack of sources, articles, notes, maybe some dense research papers, and pull out the most fascinating nuggets, making you the most informed person in the room. Today, we're really diving deep into the incredibly complex world of DNA. We're not just looking at what it is, but exploring how it's controlled with, frankly, astonishing precision, and how it can sometimes break its own rules, which is fascinating. And also, what its very ends can tell us about aging and, well, our lifespan. You've given us some really compelling material here. It peels back the layers on three quite surprising things: these hidden controls in DNA synthesis, this revolutionary idea of genetic info flowing backward, and the mystery of chromosome ends, the telomeres, and their link to how long we live. So get ready for some real aha moments. Some facts that might just make you look at your own biology in a completely new light. All right, let's start with the secret life of DNA replication. We often think of it as, a straightforward copying job, continuous. But our sources show there's this whole hidden layer of regulation, and it's not everywhere. It's focused.
That's right. And what's really critical is that initiation, just the very start of replication, is the only phase we know is tightly regulated. This makes sure each cell copies its DNA exactly once before it divides. Just once. We're still figuring out all the details, but we have some really key insights into how this is enforced.
Okay, so it's all about starting at the right time and only once. What are the molecular players involved? You mentioned DNA itself being marked.
Exactly. One major mechanism involves something called DNA methylation. Think of it as the tiny chemical tag added to the DNA. An enzyme, DAM methylase, puts this tag on a specific DNA base, adenine, but only when it's part of a particular sequence, GAT. And here's what's interesting. Our sources point out that in E. coli, which we often use as a model, the starting point for replication, OraC, is packed with these GATC sites, like 11 of them in a tiny stretch of 245 base pairs. Compare that to the rest of the chromosome, where you find maybe one every 256 pairs.
Wow, okay, so that density must be important. What happens right after the DNA is copied with these tags?
That's where the clever control comes in. Right after replication, the DNA is hemimethylated, meaning the original strand has the tags, but the brand new strand doesn't yet. And this hemimethylated ORIC gets temporarily grabbed and held by the cell membrane, sequestered. We don't fully know how the membrane does this yet, but it acts like a crucial holding pattern, a control point.
Huh. It's like the DNA needs a timeout after copying before it's allowed to go again.
Precisely, a timeout. After some time passes, the ORIC is released from the membrane, and crucially, it must get fully tagged, fully methylated by DAM methylase before it can bind to the initiator protein, DNA, again. This physically prevents it from starting replication too early.
And this DNA protein, the initiator, it's not just sitting there waiting, is it? Does it have its own clock?
It does, yes.
Yeah.
It has an internal timer in a way. DNA slowly breaks down ATP. It cycles between an active form bound to ATP and an inactive form bound to ADP. And this cycle takes about 20 to 40 minutes, so that adds another layer of timing control over when initiation can actually happen.
So putting it all together, what does this mean for you listening? It's a glimpse into these incredibly precise, multi-layered systems. They ensure your cells divide correctly. It's a fundamental process, but far more complex and elegantly managed than you probably imagined. Okay, let's shift gears. For decades, the central dogma, was the rule: DNA makes RNA, RNA makes protein. Simple flow. But our sources introduced this enzyme that just blew that wide open. It showed genetic information can actually flow backward, a real game changer.
Yes, this is the story of reverse transcriptase. It's an RNA-dependent DNA polymerase, meaning it makes DNA using an RNA template. The reverse, you find it in certain RNA viruses, specifically retroviruses. And remarkably, its existence was actually predicted by Howard Temin way back in 1962. Then he and David Baltimore independently found it in 1970. A discovery that, well, it really shook the foundations of molecular biology. Nobel Prize worthy, in fact.
So how does this backward flow actually work when these viruses infect our cells?
Right. So when a retrovirus gets into an animal cell, its RNA genome, maybe 10,000 nucleotides long, enters, along with the reverse transcriptase enzyme itself. The enzyme then does three things in sequence. First, it builds a DNA strand that matches the viral RNA. Second, it removes the original viral RNA from that new RNA-DNA hybrid. And third, it synthesizes a second DNA strand, creating a normal double-stranded DNA molecule from the original RNA info.
And this new DNA made from RNA can just integrate itself into our own genome. That sounds potent.
It is incredibly potent. That's the key. This new viral DNA often gets inserted right into the host cell's chromosomes. It can stay dormant there, hidden, or it can get activated later. Then it's transcribed back into viral RNA, making new viral proteins and genomes, assembling whole new virus particles.
It's quite the molecular acrobatics act for the virus, but is it perfect? Are there any downsides for the virus, maybe in terms of making mistakes?
That's a really important point. Accuracy. Unlike a lot of our own DNA copying enzymes, reverse transcriptases usually lack a proofreading function. They don't have that 3 to 5 foot exanuclease activity that corrects errors. And the result? A surprisingly high error rate, about one mistake for every 20,000 bases copied.
Wow, that's way higher than normal DNA replication. What are the consequences of being so sloppy, so to speak?
Well, if you look at the bigger picture, this high error rate is actually typical for enzymes that copy RNA viruses. The consequence is a much higher mutation rate, and that means faster viral evolution. This rapid change is a big reason why new strains of retroviruses like HIV, for example, pop up so frequently, constantly challenging our immune systems and treatments.
But this enzyme, even though it came from viruses, it's become a vital tool for scientists now, right?
Oh, absolutely. Indispensable. Reverse transcriptases are now standard lab reagents. We use them constantly to study DNA-RNA interactions, and especially in DNA cloning. They let us make DNA copies of messenger RNA, we call that complementary DNA, or cDNA. And that cDNA is invaluable for cloning genes, studying how genes are turned on or off, you name it.
So what this really tells us is that reverse transcriptase didn't just break a rule. It showed us biology is more flexible than we thought. It gives viruses a way to adapt quickly, but it also gave scientists an incredibly powerful tool set for genetic research and engineering. Okay, we've looked at how DNA replication is controlled, how information flow can be reversed. Now let's focus on the very ends of your chromosomes. These structures hold some really surprising clues about aging and disease.
Right, we're talking about telomeres. The name literally comes from the Greek for end part. They're these repetitive stretches of DNA sequence right at the tips of each chromosome. Their job is crucial. Protect the ends from damage, stop them from sticking to other chromosomes, and importantly, help ensure the DNA gets fully copied.
Like the little plastic caps on shoelaces, basically, preventing fraying.
That's a great analogy.
Yeah.
Exactly like that. In vertebrates, like us, the repeating sequence is TTG.
Yeah.
The other strand is AATCCC. And this TTG sequence, it actually forms a little single-stranded overhang at the very end. It's repeated maybe 2,500 times in humans at birth. But here's the thing. Telomeres shorten over time. In humans, the average length drops from about 11,000 bases at birth to less than 4,000 in old age. And it seems to happen faster in men than women.
Why? Why do they shorten? What's the basic problem that DNA copying runs into at the very end?
Yeah, that raises the fundamental issue.
Yeah.
The standard DNA copying enzymes, the polymerases, they just can't finish the job on the very end of one of the strands, the lagging strand. After the little RNA trimer that starts the copying is removed, there's no starting point, no 3' hydroxyl group for the polymerase to fill in that final gap.
So logically, our chromosomes should just get shorter and shorter every time a cell divides. That sounds bad. How do we avoid that?
It would be bad, lethal, eventually. For years, this was the end replication problem, a real puzzle. But we found out that the solution involves both the unique telomere structure and a special enzyme. That enzyme is telomerase, discovered back in 1985 by Elizabeth Blackburn and Carol Greider. Their work, along with Jack Szostak's, won them the Nobel Prize in 2009.
Telomerase, what's so special about it? How does it do what the regular enzymes can't?
The really unique thing about telomerase is that it carries its own template, an RNA template built right into the enzyme. It acts like a specialized reverse transcriptase. It finds that G-rich TKG overhang on the chromosome end, and it extends it, adding one TKG repeat at a time, using its internal RNA as the guide. Importantly, it doesn't fill the gap on the other strand directly, it just makes the overhang longer. Then, after telomerase has added enough repeats, the regular DNA polymerase can come in and synthesize the complementary strand. Problem solved.
So without telomerase doing his job, the chromosomes would just keep shrinking. Lethal, you said.
Yes. Without telomerase activity, linear chromosomes would progressively shorten each generation. If those deletions reached essential genes, the cell couldn't survive. So yes, it's absolutely essential for maintaining our genome's integrity over time.
Okay, this brings us straight to cancer and aging.
Yeah.
How does telomerase fit into those huge biological questions?
Well, connecting this to the bigger picture, think about cancer. A key feature is uncontrolled cell division, right? Immortality, essentially. And what we see in many, many cancer cells is high levels of telomerase activity, much higher than in most of our normal body cells, our somatic cells. This led to the idea maybe inhibiting telomerase in cancer cells could be a treatment strategy. Force their telomeres to shorten, make the cancer cells mortal again, and cause them to die. It's promising, but it's not a silver bullet. Some cancers maintain telomeres differently.
And the link to aging. You mentioned most of our body cells don't have much telomerase.
Exactly. That raises this important difference between our germline cells, sperm, and eggs, which pass on genes generation after generation and do have high telomerase and most somatic cells, which have little or none. When you grow normal human somatic cells in a lab dish, they only divide a limited number of times, maybe 20 to 70 times. Then they stop dividing, enter senescence, and eventually die.
And you can actually see the telomeres getting shorter as this happens.
Absolutely. There's a direct correlation.
Yeah.
Cells with longer telomeres survive for more divisions before they senesce. And just as you'd expect without telomerase, their telomeres measurably shorten as the cell culture ages.
It's fascinating that even when cells become immortal in the lab, telomerase seems key.
Yes, sometimes somatic cells in culture suddenly gain the ability to divide forever. They become immortalized cell lines. And consistently, these immortal cells are found to have switched telomerase back on, unlike the normal cells they came from. And perhaps the most compelling evidence linking telomeres and aging comes from studying people with progerias. These are rare genetic diseases causing accelerated premature aging.
Right, like Hutchinson-Gilford, where kids age incredibly fast, or Werner syndrome starting in the teens.
Precisely. Now, we don't know the exact chain of events linking the specific gene defects to aging in these conditions yet, but one thing is consistent. The somatic cells from individuals with progeria have unusually called telomeres, and they don't proliferate well in culture. This strongly supports the hypothesis that telomere shortening is a significant contributor to the aging process in humans.
So what this fundamentally tells us then is that telomeres aren't just passive end caps. They're dynamic. They're deeply connected to cell division, to cancer, and maybe to the very process of aging itself. It's a profound idea, isn't it? That the length of these tiny structures at the ends of your chromosomes could be ticking down, acting like a kind of biological clock for your cells, and maybe for you.
Wow. Okay, so we've journeyed through the intricate controls of DNA copying, we've seen genetic information flow backwards, breaking a central rule, and we've explored the incredible story of telomeres and their deep link to lifespan. Yeah, and these aren't just, separate stories. They show these amazing layers of complexity, of elegance, really, in our biology. Things that still surprise us and challenge us and definitely open up new ways to think about disease and maybe even aging itself.
So as we wrap up, what stands out to you, Hugh, from all this? Think about how knowing these intricate details of DNA's hidden life might reshape how you view your own cellular health, or how viruses evolve so fast, or even that biological clock that might be kicking away inside each of us because of those chromosome ends. The deeper you dive into this stuff, the more astonishing it all becomes.